Welcome to Module 1, Section 3. This section focuses on explaining how antibiotic resistance occurs in bacteria. Understanding more about these mechanisms should hopefully lead to development of better diagnostics and development of better antimicrobial drugs that can withstand the microorganisms' attempts to become resistant. We will start by looking at four defense mechanisms through which bacteria evade antibiotics. However, resistance to a single antibiotic may arise from several mechanisms. First, changing the bacteria to produce enzymes that inactivate the antibiotic by altering or destroying the antibiotic. For example, Klebsiella pneumoniae bacteria produce enzymes carbapenemase, which break down carbapenem drugs, beta-lactamases, which inactivate penicillins, and acetylases, which inactivate aminoglycosides. Second, reduction of drug penetration, thus preventing entry of antibiotics into microbes. The outer membrane of gram-negative bacteria is the main reason for resistance to a wide range of antibiotics, including beta-lactams, quinolones, colistins, and other antibiotics. Most antibiotics must pass the outer membrane to access their targets. For example, hydrophobic drugs can pass through by a diffusion pathway. On the other hand, hydrophilic antibiotics like beta-lactams pass through porins, and vancomycin cannot cross the outer membrane due to its structure that hinder it from using any of these passages. Any alteration in the outer membrane by gram-negative bacteria, like changing the hydrophobic properties or mutations in porins and other factors, can create resistance. For example, absence of membrane protein D2 porin in resistant Pseudomonas prevents the drug imipenem penetration. Gram-positive bacteria lack this important layer, which makes gram-negative bacteria more resistant to antibiotics than gram-positive ones. Third, acquisition of efflux pumps by expelling antibiotics from the microbes through efflux pumps. For example, some Pseudomonas aeruginosa bacteria can produce pumps to get rid of important antibiotic drugs such as beta-lactams, chloramphenicol, and quinolone efflux pump in Staphylococcus aureus. Fourth, bacteria also evade antibiotics by modifying the structure of the antibiotic target so that the drug no longer binds effectively to the target. Structural changes to these targets can prevent drug binding, rendering the drug inactive. For example, change in penicillin binding protein inhibits binding of the beta lactam drugs, thus providing resistance to this class of antibiotics. At molecular level, resistance in bacteria can be intrinsic or acquired. Intrinsic resistance is a naturally occurring trait arising from the biology of the organism. For example, vancomycin resistance in Escherichia coli. Acquired resistance occurs when a bacterium that has been sensitive to antibiotics develops resistance. This may happen by mutation that is intrinsic or by acquisition of new DNA. Thus, acquired resistance results from successful gene change and or exchange that may involve mutation or horizontal gene transfer via transformation, transduction, or conjugation. On the next slide, we will explain what mutation means. A mutation is a spontaneous event that can occur whether an antibiotic is present or not. Non-resistant bacteria exist in the process of multiplication. Some of these will mutate. Some of the mutations make the bacterium resistant to the drug. A bacterium carrying a resistant mutation has a significant survival advantage, while other susceptible bacteria will be killed rapidly in the presence of an antibiotic. The bacteria with the mutation survive, creating a resistant subpopulation. Horizontal gene transfer is the process of swapping genetic material between neighboring contemporary bacteria. This is another way by which resistance can be acquired. Transfer of DNA between bacteria contributes greatly to their evolution and adaptation, due to genes endowing their hosts with resistance to antibiotics, metals, pathogenicity, symbiosis, and metabolism of new substrates. Many of the antibiotic resistance genes are carried on plasmids, small piece of DNA, transposons or integrons that can act as vectors that transfer these genes to other members of the same bacterial species, as well as to bacteria in another genus or species. Horizontal gene transfer may occur through three main mechanisms. That is, transformation involves uptake of short fragments of naked DNA by naturally transformable bacteria. Transduction involves transfer of DNA from one bacterium into another via bacteriophages. Conjugation involves transfer of DNA via sexual pilus and requires cell to cell contact. DNA fragments that contain resistance genes from resistant donors can then make previously susceptible bacteria express resistance as coded by these newly acquired resistance genes. As explained earlier, resistance genes can be transferred from within and between bacterial species through different DNA transfer mechanisms. For example, one, resistance genes in commensal E. coli in the gut of healthy chicken can be transferred to pathogenic E. coli within the same chicken. The pathogenic E. coli, now with resistant genes, can infect human beings through ingestion of contaminated food or through infection of wounds from the environment. 2. Drug resistant non typhoidal Salmonella species and Campylobacter species in healthy chicken can be passed through the food chain to humans. 3. There is evidence suggesting that wild birds can spread resistant bacteria through migration, and that resistant bacteria can be transmitted from birds to humans and vice versa. Through further studies of the spatial and temporal distribution of resistant bacteria in wild birds, we can better assess their role and thereby help to mitigate the increasing global problem of antibiotic resistance.
What if I told you there were trillions of tiny bacteria all around you? It's true. Microorganisms called bacteria were some of the first life forms to appear on Earth. Though they consist of only a single cell, their total biomass is greater than that of all plants and animals combined. And they live virtually everywhere: on the ground, in the water, on your kitchen table, on your skin, even inside you. Don't reach for the panic button just yet. Although you have 10 times more bacterial cells inside you than your body has human cells, many of these bacteria are harmless or even beneficial, helping digestion and immunity. But there are a few bad apples that can cause harmful infections, from minor inconveniences to deadly epidemics. Fortunately, there are amazing medicines designed to fight bacterial infections. Synthesized from chemicals or occurring naturally in things like mold, these antibiotics kill or neutralize bacteria by interrupting cell wall synthesis or interfering with vital processes like protein synthesis, all while leaving human cells unharmed. The deployment of antibiotics over the course of the 20th century has rendered many previously dangerous diseases easily treatable. But today, more and more of our antibiotics are becoming less effective. Did something go wrong to make them stop working? The problem is not with the antibiotics, but the bacteria they were made to fight. And the reason lies in Darwin's theory of natural selection. Just like any other organisms, individual bacteria can undergo random mutations. Many of these mutations are harmful or useless, but every now and then one comes along that gives its organism an edge in survival. And for a bacterium, a mutation making it resistant to a certain antibiotic gives quite the edge. As the non resistant bacteria are killed off, which happens especially quickly in antibiotic rich environments like hospitals, there is more room and resources for the resistant ones to thrive, passing along only the mutated genes that helped them do so. Reproduction isn't the only way to do this. Some can release their DNA upon death to be picked up by other bacteria, while others use a method called conjugation, connecting through pili to share their genes. Over time, the resistant genes proliferate, creating entire strains of resistant superbacteria. So, how much time do we have before these superbugs take over? Well, in some bacteria, it's already happened. For instance, some strands of Staphylococcus aureus, which causes everything from skin infections to pneumonia and sepsis, have developed into MRSA, becoming resistant to beta lactam antibiotics like penicillin, methicillin, and oxacillin. Thanks to a gene that replaces the protein beta lactams normally target and bind to, MRSA can keep making its cell walls unimpeded. Other superbacteria, like Salmonella, even sometimes produce enzymes like beta lactamase that break down antibiotic attackers before they can do any damage. And E. coli, a diverse group of bacteria that contains strains that cause diarrhea and kidney failure, can prevent the function of antibiotics like quinolones by actively booting any invaders that manage to enter the cell. But there is good news. Scientists are working to stay one step ahead of the bacteria. And although development of new antibiotics has slowed in recent years, the World Health Organization has made it a priority to develop novel treatments. Other scientists are investigating alternate solutions, such as phage therapy or using vaccines to prevent infections. Most importantly, curbing the excessive and unnecessary use of antibiotics, such as for minor infections that can resolve on their own, as well as changing medical practice to prevent hospital infections, can have a major impact by keeping more non resistant bacteria alive as competition for resistant strains. In the war against superbacteria, de escalation may sometimes work better than an evolutionary arms race.